So students, I see you are back for episode 3 of this chapter. And in this part, we will be talking about the synapses, which are basically how a nerve communicates with whatever is after it. And that could be either another nerve, it could be a muscle, it could be a gland, and we'll see how that communication goes. So the neuroeffector communication, again, is the communication between a nerve and an effector. The effector is whatever is affected by that nerve. Okay, so again, it could be a muscle, it could be a gland, it could be another cell. And that junction is known as the neuroeffector junction. If it's between two nerves, we tend to name it a synapse. If it's between a nerve and a muscle, we can call it a neuromuscular junction, and so on. What would happen is that a chemical known as a neurotransmitter would be released from the nerve, and then it would activate a receptor on the effector. These receptors can either be ionotropic, so leading to changes in the membrane potential due to opening up of ion channels, or metabotropic, which would lead to changes in metabolic reactions. Here we can see, how nerves work. So there are two different things where we have convergence of action potentials or divergence of action potentials. So in convergence, we have all of these neurons are presynaptic to this one poor neuron, which would be the only postsynaptic neuron. So all of the information coming from these 4 different nerves are converging onto this one nerve. So I'll give you an example of convergence. So let's say you're crossing the street, or somebody's crossing the street, and they can hear a car, with their ear neurons. They see a car with their eye neurons, and they, let's say they can have that feeling, with these feeling neurons that they are about to get hit by a car. Now, all of that information would have to be, all that data needs to be collected and integrated together into one area in the brain. So that would be convergence, where you are collecting lots of data, integrating it, and understanding what it means. So if you don't converge all that data, it'll pretty much be unrelated data. You won't see how they're all related together. Divergence would be the spread of data to different things. So using that exact same example, now this person, their frontal lobe of the brain now understands and made the decision that it's time to run, and get away from that car. So it would have to send signals to the muscles of the lower limb. It wants to send signals to the heart to speed up, send signals maybe to their blood vessels to increase blood pressure, and so on. So you'd have to take that one piece of information and spread it over to different organs or different effectors. And we do that through a process of divergence. So this is how nerves work. They either converge all onto one area, and that again leads to the collection and the integration of data, or we diverge and spread the data among different effectors. Now the synapse is where things meet, which would be in this area, the synapse between the pre and the postsynaptic neuron. So how do these cells communicate together? The synapse can either be a chemical or an electrical synapse. So an electrical synapse, the plasma membranes are connected together through gap junctions. And remember that gap junctions are where basically we've got tunnels in the cell membranes, and the cytoplasms are interconnected together. And we said that electrical synapses are in gap junctions are very common in the cardiac muscle fibers, so that the heart can contract as one unit. So your electrical activity can go from the presynaptic neuron straight into the postsynaptic neuron. A chemical synapse is different. In a chemical synapse, there is an actual gap between the pre and the postsynaptic neuron, and we need to overcome that gap through the use of a neurotransmitter. And we'll see how that happens. In the functional anatomy of these synapses, in the electrical synapse, there really is no gap between the cells, but we have these gap junctions. In the chemical synapse, again, the pre and the postsynaptic have a little gap in the middle known as the synaptic cleft, and we overcome that cleft by neurotransmitters. So you can see here, this is what an electrical synapse looks like. This is the axon, the terminal end of the axon of your presynaptic neuron, and this right here is your postsynaptic cell. You see that the cell membranes have gap junctions in between, so these little tunnels, and the current or the ions are able to cross directly from the cytoplasm of this cell to the cytoplasm of the postsynaptic neuron. So that is an electrical synapse. And again, we have these gap junctions. We have that kind of connections in cardiac muscles. In the diagram of a chemical synapse, that's a little bit different, where this is your axon terminal of the presynaptic neuron. That is the cell membrane of the postsynaptic neuron, and we have here the gap known as the synaptic cleft. You see that the cell membranes are not touching, and that gap needs to be overcome. How is it that the electricity can go from pre to post? We have these little vesicles. The synaptic vesicles are full of the neurotransmitter. So remember, the neurotransmitter is made all the way in the cell body, and it has to be transmitted through those little proteins down to the axon terminal. We saw those videos. They are going to remain parked or docked right here. These are the vesicle docking sites. So these are little vesicles that have these are secretory vesicles full of neurotransmitter. When an action potential reaches the end of the axon terminal, certain changes happen that lead to the release of the neurotransmitter, and then the neurotransmitter pretty much diffuses or swims across the synaptic cleft and attaches to the receptor on the postsynaptic cell. Here you can see more detail, and before we go into the details, let's talk about the structure of what we're looking at. So here again is the synaptic knob of the axon terminal of the presynaptic neuron. These are the secretory vesicles. These are docked right here at the very terminal end. And we have voltage-gated calcium channels. These voltage-gated calcium channels are closed at rest. We also have the synaptic cleft, and we have the receptors on the postsynaptic cell. We also want to take a closer look at these docked or parked secretory vesicles. You can see one right here, and these are your neurotransmitters, and they are attached to the cell membrane with these what look like shoelaces, and we call those snares. And we will see what happens to these snares in order to release the neurotransmitter. So for the sequence of events that happen, the action potential reaches the end of the axon terminal. Remember, action potential is electricity, so it will open up these voltage-gated calcium channels. When that happens, calcium rushes into the cell. Calcium helps. We would have to go down here. Calcium attaches to these snare proteins and pulls those shoelaces apart from each other. That leads to the fusion of these membranes to the actual cell membrane and opening up of those vesicles, releasing the neurotransmitter in a process that we've known as exocytosis. The neurotransmitter then diffuses across the synaptic cleft and attaches to the receptors in the postsynaptic cell. Here, again, is what we talked about in writing, so we'll go over it one more time. Your axon terminals in your presynaptic neuron have your synaptic vesicles. those contain neurotransmitter molecules. That neurotransmitter could be acetylcholine, it could be adrenaline or epinephrine, norepinephrine, depending on the nerve that we're talking about. So we have different kinds of neurotransmitters, and that is what we will be talking about. The different kinds of neurotransmitters is really episode 4 of this chapter. So when the depolarization reaches the axon terminal, that'll open up the voltage-gated calcium channels. These calcium ions flow inside into the neurons. Calcium ions are attached to the SNARE proteins and activate them, and that again will lead to the release of the neurotransmitter via exocytosis. Now, as long as the neurotransmitter is there, it'll keep on activating the receptor in the postsynaptic neuron. So, in order to end the transmission and terminate the signal, we have to find a way to remove that neurotransmitter, and that can be done by different ways. Either the presynaptic terminal can actively reuptake the neurotransmitter, so it's pretty much kind of like vacuuming it or sucking it back into the presynaptic neuron. Or it could be transported by the glial cells. So remember, these glial cells are helpers. They support the neurons in their function. So to prevent overstimulation of the neurons, these glial cells can help in transport and the degradation of the neurotransmitters. Or these neurotransmitters just diffuse away from the synaptic cleft. Or sometimes there are enzymes that can deactivate these neurotransmitters. Now, what is going to happen in the postsynaptic cell? Well, this is where the graded potentials come into action. So remember we said that a graded potential is a potential that can happen at a synapse. This is what we were talking about. So we can either excite or activate the postsynaptic neuron, and that is known as an excitatory postsynaptic potential. Nobody has time to say that, so we call it an EPSP. And remember, in an excitatory or in an EPSP, you are depolarizing, which means that you are opening up sodium ion channels. But on the other hand, you could also be inhibiting. The other option would be to inhibit the postsynaptic neuron, and that is known as an inhibitory postsynaptic potential, or IPSP. In this case, you are definitely not opening up sodium channels. You are opening up either potassium ion channels or chloride ion channels. And when you do so, in either scenario, you are increasing the negativity of the inside of the cell. So instead of being -70, we are becoming -90 or -100, and that inhibits the cell. You are hyperpolarizing the cell. Okay, and that is definitely a graded potential. Remember that action potentials are never inhibitory. So an EPSP has the ability, if it reaches the threshold, it might change into an action potential. IPSPs, though, never become action potentials. So here we go. You can see it in a graph format. So your EPSP, you are opening up sodium ion channels. So the -70 is going to change a little bit. Again, if you remain underneath the threshold, you're a graded potential. If enough sodium channels are open where we reach that -55, we'll open up the voltage-gated sodium channels and a full action potential occurs. So again, your EPSPs are graded potentials with the ability to become an action potential. IPSPs, on the other hand, open up either potassium or chloride ion channels, both of which will hyperpolarize the cell. So you are going from -70 to -90 or so on. So you're going much further away from your threshold. You are inhibiting, you are deactivating that cell, and that has absolutely no chance of becoming an action potential. Now, how do these EPSPs and IPSPs interact on the postsynaptic neuron? So know that this one postsynaptic neuron gets tons of information from different cells. So you see here that all three, A, B, and C, are all three presynaptic neurons to this poor one cell. Excuse me. The green ones are excitatory synapses. So these are sending, these will lead to EPSPs, while the C1 is an inhibitory synapse. So that will cause an IPSP. How is it that so what does this poor neuron do? Does it become excited and depolarize, or is it inhibited and hyperpolarizes? Well, it all ends up on how the summation occurs. You have to sum up. So it's kind of like, balancing your pros and cons when you want to make up a decision. And so you make a list of pros and list of cons, and then you kind of balance it out and sum them together. That's what the nerve does. That's what the postsynaptic neuron does. So depending on your presynaptic messages, this postsynaptic neuron will have what is known as a summation. So that's the word summation. And all of that summation happens in the axon hillock. So remember when we were talking about in episode 1, we said that a lot of the electrical changes happen in that funnel-shaped area that we named the axon hillock. So this is what I was referring to. So we will see if the postsynaptic neuron gets one stimulus from A, that would lead to, a little tiny EPSP. If it gets another one, it gets another tiny EPSP. Neither one reached the threshold, so these are graded potentials, and it moves on. Okay, so nothing changed in that cell. But let's say we got two EPSPs from neuron A, and they were very close to each other time-wise. We call that temporal summation. So it was kind of like, instead of getting another one later on, which is this scenario right here, it got this. So they were very close time-wise. That is called a temporal summation. So they sum up, they add up, and you get a bigger graded potential than what you would have gotten if it was just one. Again, you're still subthreshold, so nothing is going to happen to that cell. Now, what happens if you get an EPSP from A and another one from B? Okay, so those are known as spatial summations because you are summing up stimuli coming from two different spots, hence the name spatial summation. So you're adding up A and B. Okay, again, you're getting a much bigger result or a much bigger graded potential than you would get if it's only B or if it's only A. Okay, but again, you're still subthreshold. What if you're getting stimuli from both of them together and they're all adding up to a lot of stimulation, so a lot of EPSPs to the point where you have reached the threshold? At that point, you will get an action potential. Okay, so again, if you add up EPSPs, you can eventually get an action potential. If you look, though, at the inhibitory synapse, the IPSP caused by neuron C, that is going to lead to a hyperpolarization, and you're going further away from your threshold, definitely zero chances of getting an action potential. If you sum up the effects of A plus C, which would be another example of spatial summation, you see that they cancel each other's effect and nothing happened to the cell. So the interaction or the result, whatever happens to this postsynaptic neuron, really depends on, how many stimuli it's getting. Are these stimuli inhibitory or are they excitatory? And again, all that summation happens in the axon hillock. Now you can see these three neurons. We have neuron A, B, and C. This synapse right here, where neuron A is presynaptic and neuron B is postsynaptic, while in this scenario, down here, the synapse here is between B and C, where B is the presynaptic neuron and C is the postsynaptic neuron. So A does not have a direct effect on C, but it can affect it through B. Okay, so if A stimulated B, B will then stimulate C. So again, this is known as the axo-axonic synapse. So you can see here an axo-axonic synapse, where neuron A has no direct effect on C, but it can affect it indirectly through neuron B. Okay, again, known as the axo-axonic synapse. So here, before we talk about the different kinds of neurotransmitters, we want to learn how can a drug affect a synapse. And because there's so much going on in a synapse, there are different mechanisms that drugs can use to lead to an effect. So for example, it could either increase, kind of prevent the storage of the neurotransmitter in the vesicles, so that now you can see the neurotransmitter is released into the cytoplasm, and that leads to its degradation by different enzymes. It could maybe destroy the snare proteins. So remember, the snare proteins were responsible for the parking or bonding or the docking of these vesicles at the axon terminal, so we could destroy these proteins. Or maybe the neurotransmitter is released and there are enzymes here in the synaptic cleft that could deactivate and break down the neurotransmitter. So we could either, activate or inhibit that enzyme. How about we increase the reuptake? Remember the vacuuming of the neurotransmitter back into the presynaptic terminal, hence kind of removing it from the synaptic cleft. We can maybe block the receptors in the postsynaptic neuron. Okay, blocking them. Remember, we have agonists and antagonists. So there are so many different ways where drugs can affect the synaptic transmission. So in this slide, we are going to talk about a couple of diseases that lead to, that affect the transmission. Okay, so we will talk about tetanus and botulism. They both have the same effect, or they both have the same mechanism of action where they destroy the snare proteins. And remember that the snare proteins were those shoelace-looking proteins that attach the secretory vesicle to the cell membrane of the presynaptic neuron. The pathogen for tetanus toxin is known as the Bacillus clostridium tetani, and the toxin is known as the tetanus toxin. Again, it is basically a protein-breaking enzyme, so it is a protease that will destroy the snare proteins, and that would lead to what is a rigid or a spastic paralysis. Okay, so there is an excess of muscle contraction. And again, that could be fatal due to the spastic contraction of the respiratory muscles. While botulism, the pathogen of botulism is known as the Clostridium botulinum bacillus, and it also destroys the snare proteins. Okay, they target the excitatory synapses. Okay, so the excitatory neurons are going to be destroyed. That is why botulism leads to a flaccid paralysis, or a paralysis with a reduced muscle contraction. We can use that. So although this is also fatal, again, due to the flaccid paralysis of the respiratory muscles, but we could use that toxin in therapeutic ways. So we can use it to treat a couple of things, including facial wrinkles. So Botox injection is basically injecting that toxin, leading to the flaccid paralysis of the facial muscles, hence decreasing wrinkles. We can also use it in cases of when patients have hyperhidrosis, which is the increase of sweating. Okay, we can inject the toxin, and that would basically lead to the flaccid paralysis of their sweat glands. We can give it to patients that have uncontrolled blinking. It helps in that condition, and in the malalignment of the eyes. And there are other diagnoses that we could use it in as well. Okay, so at this point, I'm going to ask you to stop. Okay, kind of go back and reflect on these 3 parts that we talked about before moving on to part 4.